All right, so this last section is going to talk about aerobic respiration, anaerobic respiration, and fermentation. In other words, how ATP is generated. So just a quick overview here. We have our cell. And in the cell we have a nucleus, and then we have these structures called mitochondria. And we said the mitochondria is the energy center of the cell. This is where a majority of the cell's energy is produced. So in bacterial organisms, they don't have a nucleus, they don't have mitochondria, they have this little tiny region known as the nucleoid, and that's where their DNA happens to hang out, and they're much smaller than a eukaryotic cell. So many of their reactions are just going to happen across their cell wall, but we're going to focus on how eukaryotes handle cellular respiration or producing energy. And then just know in the back of your mind, it can also apply to prokaryotic cells. They just do it in a slightly different location. So we have a molecule known as glucose. And I'm going to try to draw out the molecule here. So that's what glucose looks like. So it's a ring structure. It has 6 carbons. So carbon 1, 2, 3, 4, 5, 6. And it has oxygens in it. 1 oxygen is internal in this ring here. Now, knowing the structure of glucose is not going to be essential for this class, but some people like to visually see what's happening with this molecule. So I draw it out just to show you what it looks like. So this is what glucose looks like. And our energy production pathways usually start with glucose. This is the simplest molecule that we can produce the most energy from. So the first thing that has to happen is this glucose has to get into the cell. So there's glucose transporters that help pump glucose into the cell. Now once glucose is in the cell, we're going to start the process of glycolysis in the cytoplasm. And glycolysis is going to produce 2 molecules of pyruvate. And those pyruvate are going to go into the center of the mitochondria. In the mitochondria, they're going to go through the Krebs cycle, and then they're going to go through the electron transport chain. And eventually, you're going to end up producing energy. So let's learn about how. If you read the textbook on the process of cellular respiration, it's going to give you a very lengthy description about every single enzymatic step that leads to the production of pyruvate. For this class, you don't need to know all of the 10 steps, the 10 enzymes, the 10 substrates or 10 products, but you do need to know a few of them because there's these things called rate-limiting steps, these essential steps that take you to a certain point, and then it's kind of past the point of no return at that point. Once you get past those rate limiting steps, you can't go back. So as soon as, well, we'll start before we talk about the process here. Let's start by looking at this word for a minute. Glycolysis. Lysis means to break apart. And I've always been stressing, if you see a big term, try to break it down into its pieces. Well, lysis is a stem, means to break apart. And glyco is talking about glucose. So the process of glycolysis is literally ripping glucose in half. You're going from glucose, which has a molecular formula of C6H12O6, and you're producing 2 molecules of pyruvate, which is C3H6O3, literally breaking it in half. So from glucose, to start our pathway of glycolysis, glucose comes into the cell, and glucose immediately gets converted to glucose 6-phosphate. by the enzyme hexokinase. Now, again, the enzyme name is not going to be too important, but why does it have to be converted immediately so that glucose doesn't diffuse back out of the cell? In this class, we did a transport lab. So we had these eggs, these deshelled eggs, and we had like sugars passing into and out of the membrane and so forth. Really the water was moving into or out of the cell depending on the sugar solution concentration. If we bring sugar into a cell, that sugar wants to go down its concentration gradient. So it's going to want to leave the cell as soon as it comes in to equalize the amount of sugar on both sides of the membrane. So to try to trap it in the cell, we convert it to glucose 6-phosphate because look, now it's something new. So when we look at the concentration of glucose in the cell and glucose out of the cell, it's always going to want to come in the cell because when it comes in, it becomes something different. It's no longer just glucose. Let me add another slide in here so that I can show you what's happening with this molecule of glucose. I'm going to draw the molecule of glucose a little bit bigger. So glucose again. Looks like this. And then the hydrogens would fill up the missing spaces. So when I say that this glucose becomes glucose 6-phosphate, what I mean is, let's number all of these carbons. So the first carbon starts here, 1, 2, 3, 4, 5, 6. So if we look at all the carbons, the ring is made up of carbons with the exception of this one oxygen. Then we've got a carbon here. So that's where our 6 carbons are located for C6H12O6, glucose. Now if I say glucose 6-phosphate, what I'm doing is I'm adding a phosphate group on the 6th position carbon. From glucose 6-phosphate, it turns into fructose 6-phosphate. So now we rearrange this molecule, we open up the chain and then put the chain back together, but when we do so, fructose now looks like this. It looks a little bit more symmetrical, and instead of being a 6-carbon ring, or a hexagon, sorry, now it's a pentagon, and it looks a little bit more symmetrical, except for the fact that we have this phosphate on this 6th position carbon, and we don't have a phosphate on this carbon. That would make it truly symmetrical. So that's literally the next step that we're going to do. We're going to take fructose 6-phosphate and turn it into fructose 1,6-biphosphate. So in our very next step, we're going to add a phosphate to this guy. Now it's symmetrical. Now you can see how these two can just rip this molecule in half. So through a series of a few more steps, we end up with 2 molecules of pyruvate. So now that you've visually been able to see how this happens, it's important for you to know that for this class, the questions are going to be in multiple choice format, and it'll say, what is the function of glycolysis? When glucose 6-phosphate is formed, what is formed next? So that would be fructose 6-phosphate. Glycolysis produces 2 molecules of blank, pyruvate. Now in this whole process, we produce 4 ATP, but we end up using up 2 of them just to get this reaction to go. So we only have a net gain of positive 2 ATP. And that's not a lot of energy, but it's something. So we're on our pathway to regenerate that ATP. So now we go to the Krebs cycle. With the Krebs cycle, we start with pyruvate. Pyruvate gets converted to acetyl, which is transported by coenzyme A, so we call it acetyl-CoA. And that enters into the Krebs cycle. And we call it the Krebs cycle because as long as we are pumping in pyruvate, this thing's going to continuously go. But it's not just pyruvate to acetyl-CoA that needs to be pumped in. We can actually pump in amino acids and certain types of lipids into different entry points in the Krebs cycle. And then they can be used to make energy. We can also produce certain amino acids from the Krebs cycle. And those can go off to make structural proteins. Amino acids can be used for all sorts of stuff. So pyruvate, acetyl-CoA, and then acetyl-CoA combines in with the Krebs cycle. Now there's 8 separate enzymatic reactions, but just like with electron transport chain, I'm not going to make you memorize any of them. So oxaloacetate is going to combine with acetyl-CoA to produce citrate, and then from citrate we produce a whole bunch of other stuff. We kind of go through the cycle, and this just continues to go around. But what is important is what's given off by the Krebs cycle. First of all, in the process of converting pyruvate to acetyl-CoA, we give off carbon dioxide as a waste product. We also give off carbon dioxide as a waste product in the Krebs cycle directly. So if you think about breathing as a person, we breathe in oxygen and we breathe out carbon dioxide. Where does that carbon dioxide come from? Right here. This is where all of our body's carbon dioxide comes from. This is why we call it cellular respiration, because it's literally cells breathing. It's the exchange of gases that allows for a cell to produce energy. And these carbon dioxide molecules are waste to us. What happens if we don't breathe out these carbon dioxide molecules? They actually get converted to carbonic acid in our body. Our blood gets really acidic. Our enzymes start to misfold and denature. We slip into a coma and we die of acidosis. So that can't happen. So anytime that we have an accumulation of carbon dioxide, we start to hyperventilate to try to get that stuff out. So we breathe really fast to get as much carbon dioxide out as we can. But anyways, that's kind of a little bit above and beyond what we're focusing on here. So pyruvate to acetyl-CoA releases carbon dioxide, and the Krebs cycle directly releases carbon dioxide. The other thing that's released by carbon dioxide is these hydrogen ions and electrons. And the electrons are transferred in electron transporters known as NAD and FAD. And NAD can transport 1 proton, while FAD can transport 2 of these protons or hydrogen ions. And we also give off 2 ATP. So we produced 2 ATP in glycolysis, and now we just released 2 ATP from the Krebs cycle. So we're at 4 ATP, and we've done a lot of work so far. So carbon dioxide, like I said, is going to be eliminated as a waste. We don't want that in the body anymore. The energy, that's good to have. And these FAD and NAD carriers are going to carry this stuff to the next part. And the next step is our electron transport chain. Now, if you remember where this is all occurring in the cell, the pyruvate was brought into the mitochondrial matrix in order to do that Krebs cycle stuff. So this all happens in the mitochondrial matrix. So that's just the center of the mitochondria. So now, if you recall, the mitochondria has two membranes, an inner membrane and an outer membrane. The electron transport chain sits right here in this inner membrane of the mitochondria. We have 5 major complexes. 1, 2, 3, 4, and then the 5th one is this large turbine structure known as ATPase. So NAD and FAD carry these hydrogens to various regions in this electron transport chain. And the hydrogen ions are going to flow through, and they're going to eventually form, a concentration gradient, and they're going to start to build up inside of this intermembrane space. And as they build up in this intermembrane space, there's a lot of positive charges here, and there's a lot of the same things. So they're not happy being this close to each other, being like charges repel each other, and being that they're so concentrated. So these guys want to flow down their concentration gradient. They want to come back through this membrane. And the only place that they can do this is through this ATPase pump. Now, as they flow through, that's kinetic energy. And that kinetic energy causes this thing to rotate and rotate and rotate. And through that rotation, ADP adds a phosphate to give us ATP. And this can give us 32 to 34 ATP per glucose for a max, grand total of 36 to 38 total, if you add the 4 from both glycolysis and Krebs cycle. Now there's little electrons that are bouncing around all over the place out here, and they're going to go through these complexes and bounce, go through, but eventually these electrons have to be accepted by something. So we take these hydrogen ions plus the electrons plus oxygen to produce water. Oxygen is now known as the terminal electron acceptor. Because in the end, it accepted these electrons. It neutralized this environment so that this cycle can keep going forward. So I said that we were going to talk about aerobic respiration, anaerobic respiration, and fermentation. But so far, we've just covered aerobic respiration because we ended up using oxygen in the end as a terminal electron acceptor. And because of that oxygen, like I said, we call it aerobic respiration. But oxygen is why this is the reason why we have to breathe in. Because if we don't bring oxygen into the body, something bad can happen. If we don't have oxygen as people, these electrons and hydrogens back up. And then it's going to flood the electron transport chain, which stops the electron transport chain. And stopping the electron transport chain prevents these guys from being relieved. So these start to accumulate here, and we back up this whole cycle. Now, having excess hydrogen ions around creates for an acidic environment. So now we're denaturing proteins here and shutting down this system. So all of these enzymes that perform these 8 separate reactions are going to shut down. And this goes all the way back to the glycolysis part where pyruvate is. So instead of pyruvate being able to go into the Krebs cycle, we actually switch into this separate process known as fermentation. So fermentation, we go through glycolysis, we produce our pyruvate, and instead of pyruvate going to the Krebs cycle, pyruvate actually gets converted to an acid or an alcohol plus carbon dioxide. There's different acids that can be produced depending on the organism, but for people, this is lactic acid. So if you go do a strenuous workout and you don't properly stretch, because you know stretching opens up those blood vessels and brings more oxygen into the muscle tissue, and that helps prevent a little bit of this. But if you don't stretch properly and you don't stretch before and after and take deep breaths and so forth, and you become oxygen deprived a little bit during a strenuous exercise, what happens is you feel that burning sensation in your muscles. That's due to a lactic acid accumulation because your oxygen requirements were not met. Your body was forced to produce energy through lactic acid fermentation, and now you've got this acid in your muscles. It's not a huge worry. Like eventually you'll have to breathe, otherwise you'll die. But short-lived spurts of acid in your muscles, that acid can be easily metabolized in your liver back to pyruvate, and you can go on to make energy from it eventually, as soon as oxygen is renewed in the body. But that's what people do. We produce lactic acid, but other organisms produce all sorts of stuff. If we were yeast cells, we would produce alcohol. Can you imagine that every time you go for a jog, you get drunk because your muscles are producing alcohol from alcohol fermentation? That would be a little crazy. I think everyone would be a little bit more fit in the world too. I'm going to go for a run and get intoxicated. But anyways, so this is how fermentation works in a person. So if we do not have oxygen, this whole system backs up, and we have to do something with that pyruvate, so we just convert it to an acid. And in other organisms, they can also convert it to an alcohol. So what about the case of anaerobic respiration? Well, with anaerobic respiration, we have glycolysis, then we have the Krebs cycle, then we have the electron transport chain, and as these hydrogens are going to flow through, the terminal electron acceptor is something other than oxygen. Usually it's sulfur, and you end up producing H2S or H2SO4 or some type of compound, some derivative of this. So in anaerobic respiration, you still go through all of the same steps. You still produce the 34 to 38 or 36 to 38 ATP in the end, except you use something other than oxygen as a terminal electron acceptor. So in class, we can definitely review these processes. In lab, it'll kind of reinforce some of this with the enzyme talk and so forth. But this is how energy is produced. Glucose comes in, and as we learned, it's a catabolic reaction. So we're going to shred it into little tiny pieces, and every time we break a bond, we release energy. And that energy we want to store in the form of a proton gradient. So we rip this to shreds, and we produce this pyruvate. Then we take the pyruvate, turn it into acetyl-CoA, and break that down into tiny pieces. We release carbon dioxide and hydrogen, and then these electrons. And these hydrogens and electrons can be carried to the electron transport chain by these two shuttle buses, and those two shuttle buses are called NAD and FAD. Now we get to the electron transport chain, and these shuttle buses that carried the stuff from the Krebs cycle come and dump their garbage here in the intermembrane space. The electrons are going to bounce around between these complexes and eventually get captured by oxygen. The hydrogens are going to do something similar, except the hydrogens build up this proton gradient. So it's a whole bunch of concentrated hydrogen in here, and they want to go down their concentration gradient. So they're going to flow through this kind of like a waterfall would flow. And by flowing through this, they release kinetic energy. This thing starts to spin. And through its rotation, it takes ADP and smacks another phosphate on it to create ATP. It's physically like it's smacking them together. Like think about spinning something around in a circle and having it hit something. So it's smacking the phosphate together with the ADP, and that physical connection causes those bonds to reform and regenerate ATP. Oxygen in this case is the terminal electron acceptor. Now if as a person we run out of oxygen, we switch into fermentation. Other organisms on Earth can use other things besides oxygen as a terminal electron acceptor, and if they do that, that's known as anaerobic respiration. But people don't have that ability. People can't just suck up sulfur and use that as a terminal electron acceptor. So that's basically cellular respiration in summary. The whole purpose of cellular respiration is to regenerate ATP, and we do so through that proton gradient.